Yeah, so I'm going to be talking about some of the work that I was doing during my PhD, which was profiling the epigenome of different immune cell subsets across global cattle breeds. So this is just a bit of an outline of what I'm going to be talking about. So I'll get started with the introduction, the aims and the methods for the project. So first, just a bit of background about cattle subspecies diversification. So there are two major cattle subspecies. These are the Bos taurus taurus, or the taurine cattle, and the Bos taurus indicus, or the Indocene cattle, shown in blue and red. And so the Indocene cattle were domesticated within the Indus Valley, while the taurine cattle were domesticated within the Fertile Crescent. And from there, they've been introduced into other parts of the world and are now very global. And so further crossbreeding between these two sublineages, as well as environmental selection pressures and selection pressured by breeders, has caused further diversification between these breeds. However, most of the research so far has been focused on the European taurine breeds. And so for my project, I was interested in trying to capture some of the diversity between different breeds. So for this reason, I was studying the Holstein Friesian to represent the European taurine, Endama to represent the African taurine, and then Nelore to represent the Indocene. And so there have been some studies done looking at genomic diversity between these breeds, but there's very little research done on epigenetics. And we were particularly interested in epigenetics given its potential role in important phenotypic traits. So I probably don't need to explain what epigenetics is, but I left this slide in just to summarize the features that I was looking at in my project. So I was looking at open regions of chromatin. So this is the regions of the DNA between the nucleosomes that needs to remain accessible to DNA binding proteins, and also DNA methylation, which will be the main thing that I'm going to be talking about today. Again, I probably don't need to explain what DNA methylation is. So given that epigenetic profiles are very cell type specific, we needed to choose which cell types we were going to be studying for this project. And we were particularly interested in disease-resistant phenotypes. So I was studying 7 different immune cell types. So these were B cells, 3 subsets of T cells, so CD4, CD8, and gamma delta, and also NK cells, which all differentiate from the common lymphoid progenitor, and then also monocytes and granulocytes from the common myeloid progenitor. So all these different subsets have different functions within the immune response. And so B cells, CD4 T cells, and CD8 T cells were all involved in the adaptive immune response, while NK cells, monocytes, and granulocytes are all part of the innate immune response. And then there's also the gamma delta T cells that bridge these two systems. And so that then brings me on to the aims of the project. And so, as I mentioned, we're lacking epigenetic resources to study this in cattle, and most resources are focused on the taurine breed. And so the first aim of my project was to develop a reference methylome and chromatin accessibility atlas for the 7 different immune cell types across the 3 different breeds, which we hope can support future studies to investigate epigenetic signatures linked to desirable traits. The second aim of my project was to develop a computational approach to allow us to deconvolute the composition of whole blood samples to enable a reference-based correction for cell type abundances when doing epigenome-wide association studies using blood. And then finally, to try and improve the characterization of the non-coding genome across the bovine lineages by identifying unique epigenetic signatures. And so to address these aims, I collected blood from 3 Holstein Friesian cattle here at Roslin, isolated cells from the blood, stained them based on their cell surface markers, and then performed cell sorting to isolate my 7 different cell types. So yeah, for 7 of these, I generated attack-seq samples. So this is the technique that looks at regions of open chromatin between the nucleosomes, and also extracted RNA for reduced representation of bisulfite sequencing to look at DNA methylation. And then for 6 of the cell types, I also isolated RNA for RNA sequencing to look at gene expression. I then repeated this for 3 Nelore cattle at the University of Sao Paulo, And then again for 3 endama cattle at the International Livestock Research Institute in Nairobi. And this time we weren't able to collect samples for RNA sequencing. So this figure here is just summarizing the samples that I collected. So yeah, I had three cattle for each breed, my 7 different immune cell types. And so the numbers at the bottom here are just showing how many samples I got for each sequencing method and for each breed. So in total, we sequenced and analyzed about 150 samples. And so to check that the cattle populations that we were studying were genetically diverse, I did a PCA analysis comparing the genotypes of the sampled animals compared to almost 300 other sampled cattle of 13 different breeds, and found that the genetic relationship between the sampled cattle is largely reflective of their ancestral geographical distribution. So there is one that is a little bit of an outlier, which is the first endama cow, which looks like it could be a relatively recent cross between an endama and a European taurine. But it still looks very diverse from the other populations. So we carried on with this. So the first question we wanted to look at was, do the epigenetic profiles match known cell type specific expression patterns? So this is a heat map for different genes with known cell type specific functions. And this is showing the percentage methylation at promoters of these genes. This second heat map is then showing the accessibility of those promoters. And then the final heat map is showing the expression of those genes. And so you can broadly see that the promoter DNA methylation and chromatin accessibility and then the associated RNA-seq expression is most often specific to the expected cell type. So taking CD4 as an example, you can see that the promoter of the CD4 gene is specifically unmethylated in CD4 T cells. It's also highly accessible, and it's specifically expressed just by CD4 T cells. So this kind of confirmed that the data looked good and demonstrates that there is a close relationship between epigenetics and gene expression. And so then, having looked at differences in cell types, we then looked at differences between different cattle's subspecies. So I did a PCA analysis looking at variation in methylation profiles at individual CPG sites across all the different samples. And so this is showing PC1 versus PC2. And so in PC1, you can see that samples separate by cell type, but by looking at PC2, you can separate samples by their subspecies. So the triangles are showing the Nelore samples, which is the Indocene breed, and then the circles and the squares are the Holstein Friesians and the Endama, which are both taurine breeds. And then if we remove the effect of PC2 and just look at comparing PC1 versus PC3, we further separate samples by cell type, and this time with B cells forming a distinct cluster from the rest of the other cell types. And then finally, if we compare PC2 to PC4, we're able to separate samples both by cell type and breed. And so to look at this in more detail, we then performed a hierarchical clustering analysis based on the correlation distance between the samples, and found that samples broadly cluster by cell lineage. So you get separation of the myeloid cells from the lymphoid cells. And then within the lymphoid cluster, you get separation of B cells from T cells and NK cells. However, within these clusters, samples then cluster more by breed than they do by cell type, such that samples from the same breed are generally more similar than samples from the same cell type. So for example, a granulocyte from a Nelore cow is more similar to a monocyte from a Nelore than it is to a granulocyte from a Holstein Friesian or an Endama. And so the next question we wanted to look at was if we could use the methylation profiles for each of these cell subsets to work out the proportion of cell types within a whole blood sample. And so when performing epigenome-wide association studies, one of the big problems, and you often do this on blood because it's one of the easiest samples to collect, is that you can get changes in the cell type composition, which can lead you to identify methylation changes, which are actually just reflective of the different proportion of cell types. And so methods have been developed to try and correct for this. And one of these is a method called CyberSort X, which is what I was using. So for this, you generate your different reference profiles. You then input this into the pipeline, and you generate a signature matrix, which is basically made up of CpG sites with a unique methylation profile in each of your cell subsets. You then provide CyberSort with the methylation profile of the mixture, and it then uses the reference profiles to impute the cell type proportions within the whole blood. And so I tested this on 6 different in vitro mixtures. So these were derived from Holstein Friesian, and I just mixed 2 of the cell types at known proportions. And then I also isolated lysed blood from each of the cattle, so 3 Holstein Friesian cattle, 3 Endama, and 3 Nelore. And I determined the composition of these lysed blood samples by flow cytometry so that we had something to compare against. So in this plot, in each pair of bars, the first bar is showing the expected proportion of cell types. So as I mentioned, that was either what we know we mixed them at or was determined by flow cytometry. And then what CyberSort predicted to be those proportions. And so this is kind of a general overview, and you can see that in general, CyberSort does quite well. And this is, I should say, just using a signature matrix generated using the Holstein Friesian reference samples. So looking at how well just using those samples is still able to predict the composition of blood from Endama and Nelore cattle. So to summarize the accuracy of this, I then compared the expected proportions with the CyberSort predicted proportions and worked out the correlation values. So this is showing the results for the in vitro mixtures, in which you can see that in general it does quite well, although it does overestimate some cell types that aren't actually present within the sample. This is then looking at Holstein Friesian lysed blood, where it does much better when all the cell types are present within the sample. And then it also does very well with the Endama and the Nelore cattle predictions, even though it's a different breed. So all the results that I've presented so far have been related to CpG sites, but there are also regions of the genome called CpG islands that have an enriched population or enriched frequency of CpG sites. And these are often located at gene promoters and other regulatory elements. So as shown in this figure, they're usually unmethylated and accessible to DNA binding proteins to allow transcription to occur. And their methylation is often linked to transcriptional repression. And so we use CpG islands to investigate the link between genetics and epigenetic divergence between the breeds. And so to do this, we measured identity by state scores and compared it to methylation differences. So identity by state scores is a measure of the similarity of alleles at polymorphic sites in a genomic region between populations. So a lower IBS score indicates fewer shared alleles across the genomic region, and therefore indicates an increase in genetic divergence. So this plot is showing the identity by state scores along the y-axis, and then the CpG islands have been grouped into bins based on their methylation difference between the Holstein Friesian and the Nelore, with increasing methylation difference along the x-axis. And the green bars are showing the IBS scores between the Holstein Friesian and the Nelore. So you can see that here that as the IBS scores are decreasing, you get an increase in methylation difference, demonstrating that indeed the genetics is having an impact on methylation divergence. And so this heat map shows that this is also that there's also an association between IBS score and methylation when comparing so this is the Nelore and the Holstein Friesian, and you also have overlap with the when comparing the Nelore to the Endama, but the sites are distinct to those that are significantly enriched, significantly associated between IBS and methylation when comparing the Endama to the Holstein Friesian. So this suggests that, the genetics is having a major contribution to the epigenetic difference, and that the difference between the subspecies is a principal driver of this, potentially more than the environment. And so finally, we then looked at the relationship between the patterns of DNA methylation and chromatin accessibility at CPG islands. So as I mentioned before, most CPG islands are thought to be unmethylated and open to allow transcription to occur. And then their methylation is associated with transcriptional repression. And so to look if we saw the same thing in our data set, I performed unsupervised clustering of CPG islands based on their percentage methylation and their chromatin accessibility, and identified 6 distinct clusters. And so these are what is represented in these plots. So this first one is showing the percentage methylation of the CPG islands within each cluster. And each of the different bars is representing a different Holstein Friesian, and then they're separated by cell type. And then the next plot is showing the chromatin accessibility of those CPG islands. And then this is showing the RNA expression of the gene nearest each CPG island. And then finally, the distance of the CPG island to the nearest transcriptional start site. And so broadly, you can see that with increasing, I should say that these clusters are ordered by increasing methylation. And so you can see with increasing methylation, you generally get a decrease in chromatin accessibility and a decrease in RNA expression. However, there are some caveats to this. So for example, in cluster 3, where these CPG islands are mostly unmethylated, they're actually poorly accessible and have a very low, and associated with genes with a very low RNA expression. However, clusters 1 and 2 fit with the more traditional idea that unmethylated CPG islands are associated with chromatin accessibility and an increase in gene expression. And these CPG islands are generally located within the promoter regions. You also have clusters 4 and 5 that show very intermediate levels of methylation and are separated based on their chromatin accessibility, with cluster 4 having a higher level of chromatin accessibility compared to cluster 5. And this is then associated with a higher RNA expression in cluster 4 compared to cluster 5. And then in the final cluster, these CPG islands are mostly methylated. They're very inaccessible and they have a very low RNA expression. And these ones are the ones that are most distal to the transcriptional start sites. And so this demonstrates that the traditional thought of a binary switch doesn't seem to be true, and it's actually much more complicated than that. So that analysis was all done on Holstein Friesians, but I also repeated it for the Endama and the Nelore data. And so this is a circles plot showing the overlap in the classification of the CPG islands between the different breeds. So the ribbons show where there's overlap in the CPG islands. So those that are going to nearby clusters demonstrate that these CPG islands have very similar epigenetic profiles, whereas ribbons that are cutting across the circles plot have more diverse epigenetic profiles between the breeds. So for example, this yellow ribbon here, these CPG islands are classified in cluster 5 for the Nelore, but for the Holstein Friesian, they're classified in cluster 3, suggesting that there are some CPG islands with very diverse epigenetic profiles between the breeds. So that just then brings me on to the summary. So we found that the major difference in these samples is classified by cell type, but we also found extensive epigenetic divergence between taurine and Indocene cattle across all the cell types. We are able to use the unique profiles of each of these cell types to accurately deconvolute the composition of whole blood samples, which we hope can be used in EWAS studies to correct the cell type composition. We've shown that that elevated DNA methylation divergence at CPG islands between populations corresponds with elevated genetic divergence. And finally, we've defined 6 classes of CPG islands based on their DNA methylation and chromatin accessibility profiles. that are linked to distinct transcriptional states. And hopefully this can be used to improve the characterization of potential regulatory elements across the bovine breeds. So that just brings me on to the acknowledgements. So I'd like to thank Liam, James, and Tim, that my PhD supervisors, and then particularly, well, everyone in their groups, but particularly Andrea, who helped me with the whole genome sequencing analysis, and then everyone in all the different labs that I've worked in that have helped me set experiments up and helped me while I've been there, and all the people in bioimaging. And thank you for listening.
Sorry, I may have missed it, but what was the age of the animal when you sample?
Yes, that was one of the issues that we had. So the Holstein Friesians were quite young. They were about a year old, but because of access to different animals in Kenya and Brazil, the Endama were about 4 or 5, and then the Nelore were about 10. So there was quite a bit of difference in age.
I was wondering about the cybersort, because, you know, based on the PCAs and stuff you show, you can clearly separate B cells from other lymphoid cells and, you know, myeloid cells from lymphoid cells. But then in your expected versus predicted plots, it looks like you are misclassifying, you know, B cells, misclassifying T cells as B cells, and then misclassifying lymphoid cells as myeloid cells and vice versa, which you wouldn't, is that a worry? Like, is there a reason why this is? Because I would have thought that, you know, like if you can get such clear separation on a PCA, you should be able to, you wouldn't misclassify cells across lineages like that.
Yeah, so a lot of it is misclassification between like monocytes and granulocytes, and especially if you're comparing between the breeds, they have very different compositions as well, which can affect it. So, for example, Nelore have a lot higher percentage of granulocytes than the other cell types, and so then you get those thinking that they're monocytes, and that can skew your results.
Do you have a hypothesis about the, you see a stronger correlation of different cell types within breeds than say, like, you know, across breeds? Do you have any hypothesis for the explanation of that?
Not really. We didn't really know what to expect when we were doing that. But no, so not really.
Okay.
I might have one. Was the health status similar between the different breeds?
So they were just taken from the farm. They were just outside roaming. I did look at the blood. I did a quick, like, just looked at it under the microscope to check if I could see any parasites. I couldn't. But yeah, they should just be healthy animals out in their normal environment.
Yes, but they would be exposed to very different.
Yeah, they would be exposed to very different things. So we don't know. They could have been infected and just not showing any symptoms, but we didn't check.
Okay, thanks.
So bearing in mind, I'm an immunologist and I know nothing about what you're talking about. But in terms of that difference between lineages of cattle, could that be due to the fact that when you're doing this analysis, you're not focusing on genes or areas that are specific for lineages? Cell lineages, you're looking across the whole thing. So could you have genes that aren't associated with the lineage but are different between the subspecies of the cattle, which is why you get that effect?
Wait, sorry, I'm not sure I understand the question.
That's probably because it's a stupid question. I'll ask again another time and see if I can explain it better.
Yeah, all of this was, all of the stuff in this was that they were aligned to the taurine reference genome, which will also be skewing the results for the Indocene and Endama. So it could be repeated once we have other reference genomes.
Thanks, Jessica. I really loved your presentation. Would you say the Endama is more related to Holstein Friesian or the Nelore?
Well, it's a taurine breed, so I would assume it more to the Holstein Friesian. But it does cluster very separately on the PCA plot, but slightly more towards the European taurine, just because it's also a taurine breed. Read it out. Okay, so the question is, can you distinguish between breed type and potentially confounded environmental impacts? So because the environmental impacts are perfectly confounded with breed, we can't really, which is one of the reasons that we did the genetic analysis to try and determine what effect that is having, and showed that the differences or the association between the genetic divergence and the methylation divergence is similar when comparing the Nelore to either of the taurine breeds, whereas it's different when comparing between the taurine breeds. And considering all three are in different environments, it suggests that it's more of a subspecies effect than their environment.
So in your presentation, you saw that different component of blood system, you compare the epigenome, but I'm just wondering that which component of the immune system is more, like change in epigenome, epigenetical change in a specific environment, because you compare 3 different breeds in 3 different environments. So, and the immune cell type play a different role at different time point. So do you think that it could be a confounding factor in your study? And which cell type you saw, like mostly, in between 3 breeds, you see the more epigenetic changes?
So for the B cells, you actually see a lot of epigenetic difference even within a breed, or even within an individual. So that in terms of the B cells, you get a lot more sites that are intermediately methylated, suggesting that you need to further isolate different B cell subsets, because there's clearly a lot of variation. So that one's probably the most varied between breeds and within a population. But yeah, interesting for the deconvolution stuff. We also, because I had all the reference profiles for the Nelore, so we used that as to generate the signature matrix and then predict the other breeds, but that didn't work. So I don't quite know why it worked for the Holstein Friesian to predict the Nelore, but the Nelore couldn't be used to predict the Holstein Friesian.